Good day, this is Professor Will, CBMD PhD, and today's Wednesday, December 1st, 2021. It is 7:06 and 33 seconds, p.m. Eastern Standard Time. Today's research, childhood cancer. So let's hop to it here. The successful treatment in mice for severe childhood cancer. Date November 30th, 2021. Source, University of Gothenburg. In mice with high-risk neuroblastoma, tumors disappeared in response to a new combination treatment with precision medicines, a recent study shows. And this is a vital step toward a potential curative treatment for a form of cancer affecting young children that is currently difficult to treat. Neuroblastoma is a form of childhood cancer that affects the peripheral nervous system, that is, the system excluding the brain and spinal cord. And in Sweden, 20 to 30 children are diagnosed annually. The cancer may start in the adrenal glands, for instance, but tumors can occur throughout the body. And in some cases, the disease can heal and disappear by itself, but aggressive forms of neuroblastoma have a more unfavorable prognosis. The present treatment regimes are very hard for children to undergo, and side effects can have consequences for the rest of their lives. The study shows that a treatment combining two different precision medicines, an ATR inhibitor and an ALK inhibitor, eliminates neuroblastoma growth in mice. After 14 days treatment, the tumors had completely vanished in two independent mouse models. One of the mice relapsed after 200 days, which is a very long time for a mouse, but all the other mice are still alive. Researchers are surprised by the very positive result. It is remarkable that a 2-week protocol that combines these two precision medicines can lead to complete tumor regression in two separate mouse models of neuroblastoma. And in experiments in culture with human cell lines, researchers see the same RNA and protein expression patterns as they do in mice treated with this new regime, showing they are on the right track. And to date, the research group does not precisely understand why this combination treatment works so well. However, an important part of their hypothesis is that the mouse immune system receives signals from the tumor cells that cause the natural immune cells to infiltrate and destroy the tumor cells. The research results are beyond their expectations. They now started a follow-up with other ATR inhibitors to investigate how they affect the mouse immune system. They want to understand why this combination treatment with precision medicines against ATR and ALK is so good. The molecular mechanisms whereby neuroblastoma arise are only partly known. What probably occurs is that defects appear in the development of the PNS, resulting in an imbalance that favors formation of neuroblastoma. The underlying defects in the cell may be that an important gene has been lost, that there is overexpression and or activation of a specific protein, or that a particular protein or gene has mutated. A quarter of all high-risk neuroblastomas arise when the oncogene MYCN is overexpressed. In many cases, this overexpression occurs in combination with an increase in the activity of the ALK protein. When such oncogenes are expressed and active in a cancer cell, it tends to divide faster, and replication stress can then occur. The results from this study suggest that patients with neuroblastoma, and especially those in high-risk groups, with a combination of the MYCN and ALK genes lead to replication stress, may benefit from drug treatment with the ATR inhibitors. The drugs slow down cancer growth, but don't fix the fundamental defect. So additional treatment options are needed for high-risk neuroblastoma driven by ALK and MYCN oncogenes. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.